Welcome, everybody. Thank you for joining us tonight. I'm very pleased to introduce our speaker for tonight, Dr. John Dickson. Dr. Dickson is a neurologist specializing in Alzheimer's disease and related disorders here at Massachusetts General Hospital. He has been involved in developing the Alzheimer Therapeutics Program, ATP, for the Mass General Brigham Healthcare System. In addition to his clinical work, Dr. Dickson research focuses on understanding the changes that occur in the brain in Alzheimer's disease and other dementias. So with that, I would love to turn it over to Dr. Dickson. Thank you so much for joining us tonight.
Thanks for having me. I'm just going to share my screen now. Give me a moment while I pull that up. And so today what I'm going to do is talk about this new era of Alzheimer's disease treatment that has come about within the past year, primarily focused on the anti-amyloid therapies that have become available and first received full approval from the FDA last summer. So we're going to talk about these, talk about the program that we're implementing here in the Mass General Brigham Healthcare System, which is called the Alzheimer Therapeutics Program, which right now is focused on delivering these anti-amyloid therapies, but is intended to be flexible enough to offer additional therapies as they come available. And we'll talk about a few other treatments that are a little bit more over the horizon. So to start off with, I just want to let you know that I don't have any relevant financial disclosures. I'm going to talk about a variety of medications, some of which have been approved and others which have not been approved by the FDA. And I don't have any relationship at all with any of the companies involved in making or selling these drugs. So my interest is really in trying to provide as much information as possible for all of you so that you can understand kind of these important developments that have been happening in Alzheimer's care in recent months. So to start off, I'm going to provide a little bit of background. So Alzheimer's disease is familiar to many people, and I'm sure many who are viewing this video. And Alzheimer's disease can be a little bit confusing to talk about in some cases, in part because Alzheimer himself described two things in the original report of the disease that bears his name. What he described was the clinical syndrome, as well as the pathological findings associated with this disease. For the clinical syndrome, there are some symptoms that are well described and are part of the clinical diagnostic criteria for Alzheimer's disease. These include having cognitive symptoms, the most common of which is memory loss, but there can be other cognitive symptoms such as language difficulty, visual perception changes, difficulty with decisions or planning, and behavioral changes. And these cognitive symptoms will come on gradually. It's hard even to know exactly when they started because it's so imperceptible. And then the progression of these symptoms is very gradual as well, where it's hard to tell one day from the next how things have changed. But over time, it becomes more apparent that there is a progressive worsening of cognitive symptoms. And this ultimately reaches a point where one or more of these types of cognitive symptoms makes it difficult for patients to carry out the activities that they would normally do in everyday life. In contrast, the pathological findings are what is happening in the brain and which are seen at autopsy traditionally. There are some examples shown here, and the two key findings are the accumulation of amyloid beta, which accumulates outside of cells, and you can see some examples of that here in the brown staining, and also accumulation of tau, which occurs inside cells, particularly nerve cells in the brain that you can see in the brown here. So these two things are very different from each other, though they are unified in the Alzheimer's disease process itself. You can think of the clinical syndrome as representing the disease symptoms and the pathological findings representing at least some of the disease process that's happening in the brain. And the way that we can think about kind of unifying these particular aspects of Alzheimer's disease is that the disease process that occurs in the brain, namely the accumulation of these two proteins, as well as the other changes in the brain, like inflammatory response and cell death, occur. And that in turn leads to the symptoms that patients experience. So one of the things that's notable about the new therapies that have come about, and in particular lecanemab that's now approved, is that it's considered a disease-modifying therapy. And I wanted to talk a little bit about what that means. Traditionally, the treatments for Alzheimer's disease have been focused on the symptoms. Some of these include medications like donepezil or memantine, or some other medications that are used specifically to try to help with memory loss symptoms. But there can be medications other than these that are focused particularly on memory. For example, antidepressants can sometimes be used to help with treating the behavior changes that occur. And some treatments don't require medications. These are other types of therapies like speech therapy to help with the language difficulties, recommendations to stop driving when the visual perception changes get so advanced that it's difficult for people to drive safely, and providing assistance from caregivers, such as family and friends, for making decisions or planning things when that becomes too difficult. All of these types of treatments can help to make the symptoms less of a problem as much as possible, though they're not perfect by any stretch of the imagination. But you could imagine that perhaps one better way to approach this is to actually treat the things that are causing the disease. So that would be treating the amyloid beta or the tau or both. And the hope with targeting these, which would be considered disease-modifying therapies, is that it will actually modify the disease process so that there are fewer symptoms because the disease process has been modified. And that's what disease-modifying therapy means. And that's why this is such an exciting time in the field of Alzheimer's disease, because we have the first treatment that can impact amyloid beta and impact the disease process, which then leads to a change in the symptoms that patients experience. So now I want to talk specifically about anti-amyloid therapy, which is this type of therapy that targets these amyloid plaques in the brain and helps remove them. So to explain a little bit about why amyloid was considered as a target and is the most advanced therapy that has now even made it into clinical practice, I wanted to share this, which is called the amyloid cascade hypothesis. And this is the idea of how the Alzheimer's disease process works. It's largely based off of some of the genetic changes that occur in rare cases of Alzheimer's disease that change the amyloid beta in those patients, that makes it more prone to aggregate and deposit in the brain. And then subsequent to that, there are other changes that occur in the brain, such as changes in the inflammatory response, injury to nerve cells, the development of the tau tangles, and then ultimately leading to cell death and the symptoms of dementia. So since the amyloid problem occurs early, one of the thoughts is to target this so that you can modify the rest of the downstream cascade. Anti-amyloid therapy in its current form is made of antibodies, which are like the antibodies that the immune system uses in the body to fight off viruses and bacteria primarily. But these antibodies are directed against amyloid beta protein itself. And these antibodies help the immune system remove the beta amyloid from the brain. So the way this looks is you have an antibody that binds to the amyloid plaques, and these help the immune system of the body remove these plaques from the brain so that it's largely cleared out of the amyloid beta aggregates. Because these antibodies are not absorbed well in the stomach, it has to be given by an IV infusion, which is one less convenient aspect of this type of therapy. It requires people to go into an infusion center. Overall, these antibody therapies have been tested in mild cognitive impairment or mild dementia stages. So if you think about how cognitive symptoms occur, there's a spectrum. People can have normal cognitive function. They can have subjective cognitive decline, which is symptoms of memory and thinking changes without any abnormalities on cognitive testing. And then mild cognitive impairment is where the symptoms become prominent enough to be reflected in testing like cognitive tests that are done in office or a neuropsychological evaluation. And then dementia is the stage where patients have difficulty doing the tasks of everyday life. And this is split in the mild, moderate, and severe categories. So the anti-amyloid therapies have primarily been tried so far in this box here, which is the mild cognitive impairment or mild dementia range. I will say that there are some clinical trials that are pushing further back, that are ongoing, that are actually taking patients who have normal cognitive function or subjective symptoms and trying to prevent progression at that time point, though that's all still experimental. In terms of the side effects of this therapy, all of the anti-amyloid therapy in general has the risk for these side effects called amyloid-related imaging abnormalities, and these are seen on MRI. There are two types, one which is brain swelling and another which is brain bleeding, and we'll talk about that in more detail a bit later. Another side effect that can happen is an infusion-related reaction, and this is kind of like an allergic reaction of sorts or an immune reaction to the antibody as it's being infused or shortly after it's been delivered through the IV, and we'll talk more about that as well. Lecanemab is particularly notable among the anti-amyloid antibodies because it's the first to receive full FDA approval, and that has allowed it to enter clinical use in the United States, and that includes at Mass General and also the Mass General Brigham healthcare system. So we do have patients who are receiving this therapy currently within our healthcare system. This particular antibody is given by IV every 2 weeks. And the reason that we know about how it works and what the side effects are based off of this phase 3 clinical trial that was conducted and reported in the New England Journal of Medicine at the beginning of last year. So this was a very high quality study where it was double-blind, randomized, and placebo-controlled. It included over 1,700 participants throughout multiple different centers and included patients with mild cognitive impairment or mild dementia, as we discussed. And there was a requirement that the amyloid that's depositing in the brain be identified either through a spinal tap with analysis of the spinal fluid for the amyloid protein or an amyloid PET scan that allows the amyloid to be visualized on the scan. And the length of the trial was 18 months. So this is the main effect of lecanemab on cognitive function. It uses a scale called the Clinical Dementia Rating Scale, and this is kind of a reflection of overall cognitive ability, including aspects of cognitive function as well as functional status in terms of activities of everyday living. And you can see here the blue line is the placebo line. This is not active drug. And then the orange line is the lecanemab group, followed over the 18 months. And something that you can recognize is that both groups had a decline in their cognitive function. So this demonstrates that lecanemab does not completely stop cognitive decline. It doesn't reverse cognitive decline. It simply slows it down. And you can see that by the difference in the slope between the lecanemab group and the placebo group. And at the end of the 18-month trial, there was about a 27% reduction in cognitive decline between these two groups. Another way to think about this is the time that was delayed in terms of the cognitive changes. So if you look at this point here in the lecanemab group and draw the line over to the placebo group, you can see that there's about a 6-month difference between them. In kind of the actual crunching the numbers, it's between a 5 and 6 month difference or delay in cognitive change over the course of the 18-month study. In terms of the side effects, we talked about one, which is called the amyloid-related imaging abnormalities, or ARIA. And I mentioned that there were two subtypes. One is swelling in the brain, which is called ARIA-E, and this is an example of that here. You can see this light gray area here or here, and that's area of swelling in the brain that's occurred after someone has received an anti-amyloid therapy. This can also occur where the swelling occurs slightly outside the brain, and this is sort of laying on top of the brain in one of the folds where there's some fluid accumulation and swelling there. The other subtype is called ARIA-H, which is bleeding in the brain. And in most cases, when this occurs, it's very small areas of bleeding, perhaps even microscopic, but can be seen on MRI. And these little black dots are kind of examples of what that might look like. Sometimes it can occur on the surface of the brain as well, such as what you see, this little black line here. That's blood that's kind of just on top of the surface of the brain. And both of these are considered ARIA-H. In many cases, it is relatively small areas of bleeding, but it can progress to more significant areas and even become what would be considered a hemorrhagic stroke. It's important to recognize that ARIA can have a wide range of clinical manifestations, and some patients are asymptomatic. They don't even know that it has happened. The only way we know about it is that an MRI showed it. And sometimes there can be very severe manifestations like the hemorrhagic stroke that I talk about, or seizures. So in terms of what the symptoms overall can be, there's a wide range. Some of them can be relatively mild and nonspecific, like a headache, and others can be quite serious, like stroke-like symptoms, seizures. So there's a very wide spectrum, and sometimes it can be difficult to tell if a patient is having a headache simply because they have migraines and they're having their regular migraine headache, or if it could be a symptom of ARIA. So an MRI is often needed to kind of distinguish between those possibilities. In terms of the risk factors for developing ARIA, receiving anti-amyloid antibody therapy is a risk factor, as you might expect, since this is what kind of led to the identification of this particular condition. This is not relevant for lecanemab, but in some cases, different doses of anti-amyloid antibodies have been used, and the higher the dose, the higher the risk for ARIA. The risk for ARIA is highest in the first 6 months of treatment, and it's associated with a particular gene and a particular version of that gene. The gene is called ApoE. It's involved in cholesterol metabolism, and there's something called the E4 allele, which is one of the variants of the ApoE gene. It's not exactly a mutation, it's just a genetic variant, and this the presence of this particular variant increases the risk for developing ARIA. And then cerebral amyloid angiopathy, which is a condition where there are many microbleeds that occur in the brain, is also a risk factor. In terms of the lecanemab trial, there was ARIA that was identified, and I'm not going to go through this in great detail due to the time, but you can see that for ARIA-E, which is the swelling, 12.6% of patients experienced any type of ARIA-E, whether symptomatic or asymptomatic. And 17.3% of patients treated with lecanemab developed ARIA-H, which is the areas of bleeding that occur. So it's not, a majority, but it's also not a very, very small amount. So it's important to take this into consideration when prescribing lecanemab and deciding whether or not to receive treatment with lecanemab. Whether ARIA occurs or not can impact the plans for lecanemab therapy. So the severity of ARIA can be determined based off of clinical symptoms as well as the radiographic appearance on MRI. And there's a recommended scheme for dosing where if there is no symptoms and very mild appearance on the MRI, it's possible to actually continue treatment with regular monitoring of MRI without any interruption in therapy. If the symptoms are mild or moderate, or if the radiographic severity is moderate, then the recommendation is to suspend dosing, where there would be a temporary pause on lecanemab infusions and routine monitoring of MRIs every month until the changes on the MRI have resolved or stabilized. And then if there's very severe clinical symptoms or severe appearance on the MRI, the recommendation is to discontinue the therapy completely. The other side effect is infusion-related reactions, and these are kind of like immune or allergic type reactions. There are a variety of symptoms such as chills, aches, shakiness, joint pain, nausea, vomiting. You might recognize these as sort of flu-like symptoms, and that's kind of what they are. It's probably like a release of kind of inflammatory messengers, similar to what you have when you have a cold or a flu. But in some cases, people can have an allergic reaction, which can cause, more serious symptoms like anaphylaxis, where the blood pressure could go low. There can also be high blood pressure or change in oxygen level if there's difficulty with breathing. About a quarter of patients in the lecanemab trial had an infusion-related reaction, and most occurred within the first infusion. So if it's going to happen, it usually happens early. Most of them were mild or moderate in severity, and very rarely were they severe enough to require that the infusions be stopped completely. The way that the infusion-related reactions are treated is that the infusion can be slowed down or even stopped if necessary. And the treatment of symptoms can be with something like acetaminophen, especially for like chills and fever, headache, antihistamines for an allergic type reaction, and steroids for a very serious allergic or immune reaction. These treatments can be given as needed after the symptoms develop, the first time they happen. But if we know that those have happened in a patient, in subsequent infusions, pre-treatment can occur where patients are given the treatment that they required ahead of time to try to prevent or reduce the symptoms with subsequent infusions. So now I want to talk about the Alzheimer Therapeutics Program, which is the program that we have in the Mass General Brigham healthcare system that is designed to help us deliver lecanemab therapy currently and eventually, hopefully, more therapies for patients with Alzheimer's disease. So this program is a bit different from a lot of the outpatient clinical care models that you may be familiar with, such as your primary care office, a neurologist or cardiologist, or any other specialist you may see. Instead of seeing one particular specialist on a longitudinal basis, this is more of a team-based approach, similar to the way that care is delivered on the inpatient service, where there's a team of people who are involved in the care and they rotate on and off the service on a regular basis. This particular program is focused on lecanemab for now and eventually other Alzheimer's therapies, hopefully. And we will be involved in both the administration and safety monitoring of that therapy and deciding about if it needs to be stopped or paused, or how to respond to possible side effects. Because of the intense evaluation and monitoring required at this time, we're not able to manage the other aspects of dementia care, such as other memory medications, recommending community services, things like that. So it is important that patients have another provider, such as the person who refers them to the Alzheimer Therapeutics Program, that can be a resource for patients for the non-lecanemab related care. For patients who are referred by someone who feels that they don't have the experience to be able to manage the other aspects of care, it is still possible to place a referral to cognitive and behavioral neurology or memory disorder specialists for kind of comprehensive cognitive evaluations and ongoing management. This program is developed by experts across the Mass General Brigham system, particularly those at Brigham and Women's Hospital and Mass General Hospital. And we have harmonized procedures, though the actual orders and physician pools are distinct for now, but hopefully we'll have more integration as time goes by. The aspects that the program handles are screening and eligibility determination for patients to make sure that they are reasonably likely to benefit, but make sure that they don't have too high of a risk for developing side effects. With regard to side effects, monitoring for the safety, to try to minimize the side effects or to respond quickly when they do occur, and to be available quickly for urgent or emergent issues that require prompt attention. The delivery of lecanemab therapy is quite complex and requires a coordinated team of individuals, including neurologists, geriatric psychiatrists, pharmacy, radiology, infusion staff, and all of the staff that are involved in making those programs work. So it requires quite a bit of coordination. And this program is sort of like the conductor of the orchestra, if you will, trying to make sure that all of that runs as smoothly as possible. And finally, we are trying to collect information about these medications, both for internal quality improvement as well as research efforts. And since this is a very new medicine, being able to learn more about these drugs and the side effects that they have can be very helpful for helping us improve this program and improve care, not only for our patients, but also patients across the country and across the world. So you may be wondering how you could get involved with the ATP, and that requires a referral from a healthcare provider. That can be a primary care doctor or a specialist. And there are some kind of basic requirements that we ask referring providers to make sure that patients meet, just to help us make sure that the evaluations are as efficient as possible. The requirements include patients that have a mild cognitive impairment or mild dementia, who've had a brief cognitive assessment performed in the office in the past 6 months, an MRI of the brain performed within the past year, not on a full-strength blood thinner because of the bleeding risk that we talked about, and a plan to have continued follow-up with a referring provider to provide the longitudinal care that the ATP cannot provide. Then once patients are referred, there is an eligibility evaluation, which involves looking over some of the information that's already in the medical record and often requiring some additional testing to be done. The goals of this eligibility evaluation is twofold. One is to make sure that there's reasonable likelihood that patients will benefit based off of the information that we have from the clinical trial, and also trying to minimize the risk of side effects. And with any medical decision or intervention, we try to weigh the risks and benefits and make a reasonable decision based off of that, and work to guide patients in making those types of decisions. So this really reflects that type of decision-making model. The components that are required for this evaluation include a clinical assessment of symptoms and their severity, memory and thinking testing, which can be done in the office with things like a MoCA test or MMSE, as if you may have heard of those in the past. A medication review to make sure that there are no medications that might be dangerous. For example, full-strength blood thinners due to the bleeding risk associated with this medication. A review of the medical history, because there are certain serious medical conditions that may be difficult for patients to undergo treatment for that, as well as treatment with lecanemab. An example of that would be patients who have cancer under active chemotherapy. It can be difficult to know which side effect may be coming from which treatment, and there are other reasons that cancer and or chemotherapy might interact in a negative way with patients who are receiving lecanemab. We also want to do blood work within 6 months prior to the evaluation to make sure that there are no major problems, such as a bleeding disorder that might increase the risk for bleeding complications. We also require an MRI that's done within the previous year to rule out significant prior bleed or a severe vascular disease. And we do require evidence that there's amyloid beta deposits in the brain, because if they're not, this medication is not going to do any benefit for patients. And the way that that can be demonstrated is either with a spinal tap or an amyloid PET scan. And finally, genetic testing of the ApoE gene is necessary, because that helps inform about risk for developing ARIA and helps patients make informed decisions about their risk for developing ARIA to help them decide if they want to proceed with therapy. So here's the pathway to the lecanemab treatment in the ATP. It starts off with a referral from either the primary care doctor or a specialist. Then there's a triage process, and then a decision about if there's additional testing that's needed, in which case there's an initial clinic visit where some preliminary assessments are made, and then the additional testing is ordered. Once all the testing is available, then there's a clinic visit to review eligibility and to order lecanemab if that's appropriate. Once that happens, there is some insurance review and paperwork that needs to happen in some cases. And then finally, there is the infusion at the infusion center. And I've drawn it as kind of a linear process, but I do want to just make it clear that depending on how the evaluation turns out, not all patients will progress to the next step if they don't meet the eligibility criteria. And I know that that is disappointing for many patients, but we are trying to make sure that we deliver this in a safe way. And I just wanted to say a little bit about what happens if patients are found not to be eligible for lecanemab. They should certainly follow up with the referring provider for ongoing care. And if necessary, that provider can consider a referral to the memory disorders unit or dementia specialist in order to have expert care with the medications and therapies that are available besides lecanemab. Another option is to consider clinical trial participation. That's a full discussion that we won't get into in great detail here, but it is a way for patients who may not be eligible for lecanemab to find other possible treatments that, in this case of clinical trials, would be experimental, but could give early access to cutting-edge therapies. This is the Massachusetts Alzheimer's Disease Research Center website, and there's a tab that has research, and if you go to that, you can look for clinical trials that are ongoing that you might be eligible for, or you can talk to the folks in the ATP if you're interested in finding out more about clinical trials if you are deemed ineligible for lecanemab. Safety monitoring is a very important part of the ATP because we do want to make sure this is done as safely as possible. For that, we have an observation period that happens after every infusion to monitor for infusion-related reactions. It's a longer period early on and then gets shortened as time goes by because there's less risk of a new infusion reaction later on in therapy. There's routine monitoring with MRIs for all patients prior to the 5th, 7th, and 14th. Sorry about that. That should be 14th infusion. And this is based off of the clinical trial process. An MRI can also be obtained at any point if necessary, if there are any concerning symptoms suggestive of ARIA. And then we have routine clinic follow-ups every 6 months as another way to check in and make sure that everything is going okay. And then finally, we have an ATP clinician on call who's available by page 24/7 in case there are concerning symptoms. This is an overview of what a typical experience would be like. In the prior year before starting therapy, the MRI has to be done. There's going to be some initial evaluations with lab work and other testing that is, ultimately supporting the decision about eligibility. And then there would be a visit in the clinic where the review of eligibility occurs. Patients have to sign an informed consent for the therapy. And for patients who are covered with Medicare, we'll talk about this in more detail later, but there's a requirement for a registry, so we capture the information required for that at that visit as well. Then patients can have their first infusion once they're scheduled in the infusion center, and then they'll have infusions every 2 weeks, which are not shown here because that would be way too overwhelming to see. And then there are these MRIs that occur as kind of standard protocol for everyone, as well as additional MRIs that may be needed if there are symptoms, as well as follow-up visits every 6 months for kind of routine check-in to make sure patients aren't experiencing symptoms that haven't been reported, and also to support the data collection for the registry requirement that Medicare has and some other insurers. And then finally, once you get to the 18-month mark, that is theoretically the last infusion based off of the clinical trial. There is some uncertainty about whether or not that should be continued or not. And unfortunately, because this is when the trial ended, we don't have empiric data to guide those decisions. So I want to touch on the insurance coverage issues because that's an important part of this as well for patients and their family. The Medicare policies are the most clear, and it's the simplest process because there's no prior authorization paperwork. There is a requirement to participate in a registry, which is kind of a simple de-identified data collection that I'll talk about more in a moment. For commercial insurance, which is basically non-Medicare, non-VA, the policies are generally more complex. Each plan has a different policy. Many of them require prior authorization paperwork to be completed. Some require registry involvement, others don't. And unfortunately, it's difficult to know the exact details of coverage unless the lecanemab has been prescribed and the process has gone through with working with the insurance based off of that. This is an area that I hope will receive more clarity and more streamline in the future, but this is kind of the reflection of reality right now in the early days of this therapy. For Medicare specifically, for those who have kind of the original Medicare and not a replacement plan, the coverage is the standard 20% coinsurance for the Medicare approved costs of lecanemab. The published cost of lecanemab for a given year is $26,500. So 20% of that would be $5,300. And this is covered by Medicare Part B, which is the medical part of Medicare. And there's an additional deductible that has to be met before the reimbursements occur. There are some other plans within Medicare, such as Medigap, secondary insurance coverage, or Medicare Advantage. All of those have different requirements, and the plan would have to be contacted individually because it wouldn't necessarily follow with kind of the traditional original Medicare policies. The Center for Medicare and Medicaid Services, or CMS, has a requirement that patients participate in a registry, and this is required for coverage. There has to be participation in an approved registry. Medicare has their own, but others can be proposed as well. We're currently using the Medicare registry, and there's really not much extra you have to do for this. It's all handled by your care team. This requires patients to be receiving lecanemab, have regular follow-ups every 6 months. There are some test scores that are done primarily just in the course of routine clinical care. So we would just be reporting those to the Medicare registry. This is de-identified, so it doesn't have your name, date of birth, social security number, anything like that. And then this is handled by the prescribing physicians or staff in their office to put the data into the registry. I also wanted to mention Eisai Patient Support Program. Eisai is the manufacturer of lecanemab, and they have a website here for their support program that can be useful for patients, especially those who don't have Medicare and have more complex coverage situations. They have patient navigators that can help with doing an assessment of coverage and identifying the programs that Eisai has that might help to make up for differences in coverage. So if patients are having difficulty with insurance coverage, this is a great resource. So now I want to move on to some highlights on emerging potential treatments. These are things that are still experimental, not approved for use, but I thought would be useful to talk about as we kind of think about moving beyond lecanemab in this new era. The closest thing to the clinic along these lines is called donanemab. It's another anti-amyloid antibody that's similar to lecanemab, currently under FDA review. One aspect of this that's beneficial is that it requires only monthly infusions as opposed to every 2 weeks. It's also been studied in a phase 3 clinical trial, very similar in a lot of ways to the basics of the lecanemab and donanemab trials, though there are some differences that I'll talk about. These are the effects on cognitive function. There are a couple shown here. They're both different ways of measuring cognitive change, and you can see that in both cases, as with lecanemab, there was a continued decline, only less so in those treated with donanemab compared to placebo. This is the exact same outcome measure that was used in the lecanemab trial that I showed. You can see kind of a similar trend where the slope is less for donanemab than placebo. And there's, again, a 28.9% reduction as opposed to 27%. So very similar in terms of that, comparison. If you look at the time difference, you can again see that if you draw a line between the endpoint here with donanemab and where people are with placebo, it's about a 6-month difference over the 18 months of this study. Donanemab side effects are similar in general to lecanemab as well. In particular, there's ARIA-E, which is 24%, and ARIA-H, which is 19.7%. I'm not going to go through all the other details, but it's important to recognize that ARIA is still a consideration for donanemab. There were some innovative ideas in the donanemab trial that were not used in the lecanemab trial. One of those was using repeat amyloid PET scans to determine treatment response and guide decisions about treatment. With lecanemab, there was one PET scan if you had it before treatment started, and that was it. And then you just continued on for 18 months regardless. With donanemab, people had repeated amyloid PET scans, one before treatment and then periodically after treatment started. And if there was evidence of clearing of the amyloid on the PET scan, the treatment was stopped because it was felt that it was no longer necessary since it had already removed all of the amyloid. This donanemab trial also used a tau PET scan, which uses a PET scan that looks specifically for the tau protein as opposed to the amyloid protein, and using that tau PET response to predict the response to therapy. And it was found that donanemab was more effective in participants who had lower tau burden. So that could be useful in helping decide who might benefit most from the treatment with donanemab. So just to compare the two antibodies, lecanemab is the one that's currently available. Donanemab is the one that is still pending FDA review. The dosing is different. Lecanemab is every 2 weeks. Donanemab is every 4 weeks. In terms of demonstration of amyloid deposits, lecanemab used either spinal tap or amyloid PET. And for donanemab, it was only amyloid PET. For tau PET, that was not used in the lecanemab study, but it was used in donanemab. And lecanemab did not have any repeat PET imaging to guide therapy to show clearance of amyloid, but donanemab did. So Up to this point, it's seeming like perhaps donanemab might be a little bit better. But when you look at some of the downsides, it flips. So the rates for ARIA-E, which is the swelling that occurs as a side effect, there's about half the rate in lecanemab compared to donanemab, though the ARIA-H, which is the bleeding complication, is a similar percentage. And then the body can actually develop an immune response to the drug antibodies, where the body kind of recognizes those treatment antibodies as foreign and develops an antibody against them. That occurs in about 40% of patients with lecanemab and 92% with donanemab. And this is important because that can provoke a bit more of an inflammatory response, infusion-related reactions, and it can also impact the efficacy of the medication because the anti-drug antibodies are removing the treatment from the body and not allowing it time to remove the amyloid. So in that sense, lecanemab is better in terms of the downside. So if donanemab does become approved, then there's going to be some challenging decisions about which one would be right for which person. So as I mentioned, donanemab is currently under review by the FDA. The decision from the FDA was expected earlier this year, but then in March, the FDA informed Eli Lilly, who's the maker of donanemab, that it was going to convene an advisory panel to discuss the phase 3 trial and determine or make recommendations on how the FDA should decide about providing approval or not. The goal of this advisory committee is to further understand the safety results in the donanemab-treated patients and the efficacy implications of the unique clinical trial design, which is what I mentioned earlier, including the limited duration dosing regimen that allowed patients to complete treatment based off of the assessment of amyloid plaque and the inclusion of participants based off of tau levels. There's no further information about when the advisory committee is going to meet, and the decision from the FDA is dependent upon that advisory committee's recommendations. So it's still unclear what to expect from that and what the timing will be. There's another treatment that I wanted to highlight. This is a tau-targeted therapy as opposed to amyloid. It's called Ionis Mapped RX, or BIIB080, which rolls right off the tongue. So I'm going to say Ionis Mapped RX instead. As I mentioned, this is a tau treatment as opposed to an amyloid therapy. And it's called an antisense oligonucleotide is the way that it works, and it blocks tau synthesis. And we'll talk about more about what that is. The overall effect of this drug is to reduce tau levels, which you might think would be useful given that tau accumulates in Alzheimer's disease. One notable thing about the way this is delivered, as opposed to an IV, this is given by what's called an intrathecal injection. And that's essentially a spinal tap and then injecting the medication into the spinal fluid. And in the initial trials, it was dosed every month or every 3 months. The phase 1 trial has been completed, and I'll show you some of those results. And the phase 2 trial is currently underway. So this is how this Ionis Mapped TRX works. It affects the way that proteins are made in the cell. And what happens in a cell to make a protein is the cell uses DNA to make RNA, and then the RNA to know how to make the protein. So as an analogy, it's often said that the DNA is like the blueprint of life, whereas protein is the building block of life. But in that analogy, it's a little hard to know what the RNA is. So I want to provide you with an example of how to kind of understand this. So let's say you're looking at your blueprint and you see this word here, laterum, I believe. I must say that my Latin is a little bit rusty. So I am not sure what this means. So I'm going to have to use a translator, maybe a dictionary or Google Translate or something. And it turns out that that is the Latin word for brick. So now I understand what brick is, and I know that I need to make some brick in order to build the things that the blueprint requires. Putting that to the example of tau, the tau gene, which is called MAPT, and that's where the MAPT in MAPT-Rx comes from. That's the DNA that helps the body know how to make tau. It's translated in a way that the cell can know how to make the protein in tau RNA, and then that leads to the body making the tau protein. So the way that the antisense oligonucleotide works is it's kind of like a sheet of paper that gets put over the translated blueprint so that you can't read it anymore. And since the body can't read it anymore, it doesn't know how to make the protein. So this antisense oligonucleotide blocks the ability of the cell to use the DNA code to make protein, and that lowers the levels of the protein. Now, in this example, I've removed all the proteins. That's not the case in the body because it's not 100% efficient. But you can see in some of the phase 1 trial results that in spinal fluid with multiple treatments, over time, the levels go down at the higher doses, and at the lower doses, it goes down somewhat and then rebounds after the treatment is stopped. So this is a proof of principle that this treatment does lower the tau protein levels as expected, and at higher doses, there's a higher and more sustained response. You can also see this in tau PET scans, which uses brain imaging to look at the levels of tau in different regions of the brain. And in this color-coded scale, the red is a lot of tau and the blue is very little. And you can see that in the treatment with a fairly high dose, you can see that in many cases, there's less red as time goes by. And that's true for most of these. It's not true for all. You can see, for example, this one, there's actually an increase in red. I'm not sure what happened there, but this person did not respond very well to the therapy. But overall, you can see that even in patients who have a lot of red, that it will diminish in many cases. So this is again demonstrating that this therapy works to reduce tau levels. This study was not intended to determine about cognitive outcomes, though. That will be subsequent studies. In terms of side effects for this medication, a lot of them tended to be related to the fact that a spinal tap or LP had to be done. Sometimes people can have a headache from that, procedural pain. Some people experienced other kind of non-specific symptoms. These are small numbers, so it's hard to know if it's related to the treatment or not. But importantly, there's no ARIA because this is not targeting amyloid, this is targeting tau. So this is an interesting treatment in part because it could be useful in patients who can't receive lecanemab or other anti-amyloid therapy because of the ARIA risk, but otherwise may need treatment. This could potentially be an option. Another consideration, if this type of therapy targeting tau does work, would be combination therapy in the future, which I think could be a good approach, at least given the understanding that both proteins are accumulating in Alzheimer's disease. So I want to wrap up with some take-home points for you. We talked about anti-amyloid therapy, particularly that this is the first disease-modifying treatment for Alzheimer's disease that's received full FDA approval, and lecanemab was that anti-amyloid therapy that received full approval from the FDA. It's now in general clinical use here in the United States. The effect of these treatments is to slow cognitive decline, though it doesn't stop it or reverse it. And there are some notable side effects, including ARIA, which is areas of swelling or bleeding that occur in the brain, or infusion-related reaction. We also talked about the MGB-ATP, which is the clinical program that is designed to deliver new therapies for Alzheimer's disease. And we talked about the evaluation process and monitoring that occurs for lecanemab specifically. And we discussed that this is a team-based care model because of the complexities of treatment. We also talked about some notable therapies for the future, including donanemab, which is another anti-amyloid therapy, and Ionis MAPT-Rx, which is a tau lowering agent. So with that, I just want to acknowledge the people who have played a key role in developing the ATP at Mass General and Brigham and Women's Hospital. And I just want to acknowledge that it's taken a lot of work from these people over many years to make this possible. So now I will open it up to time for questions and discussion.
All right. Thank you very much, Dr. Dickson. That was a fantastic presentation. I see some questions already in the chat, but as we go along, if you have a burning question in mind, please feel free to type it out in the chat as we go along. I will try to do my best to get through as many as we can with the time that we have left here. Okay. So first question here on the docket, it comes from Jack, and Jack asks, is there any research to determine if post-hypnotic suggestion given early in the disease can help control unwanted behaviors later in disease? If post-hypnotic suggestion can possibly help the patient recall information from a part of the brain not yet impacted?
I'm not aware of specific evidence in the literature about that, though I have to say that I haven't looked in great detail on that topic.
Okay. Jack also asks, and I think this was covered during the presentation, but just to kind of reiterate, does Medicare and Advantage plans cover this medication? I believe it covers, what, 20% of the total cost after the deductible is met?
Yeah, traditional Medicare covers 20% after the deductible. Medicare Advantage is often contracted through another insurance company, and that would be related to the policies that that insurance company has. So it may vary from the kind of traditional Medicare policy.
All right. Next one comes from Adriana, and Adriana asks, is this medication, I'm assuming, I believe it was referring to lecanemab, is lecanemab available worldwide, or is it exclusive to the United States?
It came to the US first, but I believe Japan has now approved it. I'm not sure about other locations in the world. I think it is being evaluated in Europe, but I'm not sure if it has been fully approved yet.
Okay. Daniel asks, what do you think about Vindamax?
I'm not sure of what that is.
Danny, if you want to give some clarification or some additional information, be happy to revisit the question. I just don't know if we have enough information to go on for Vindamax. All right. Ash has the next question, and Ash says, if somebody gets the side effects of ARIA-E or ARIA-H, will it resolve, or are you stuck with the effects of that side effect forever?
So it depends a bit on how severe the ARIA is. In general, for ARIA-E, we would expect that it would resolve over time as long as the treatment was discontinued with lecanemab, especially if it were a more severe reaction that we see on the MRI. You would probably stop the therapy, and then the ARIA-E over time would resolve. Sometimes if it's very severe, patients would require treatment with steroids to calm down the inflammatory response. In that case, for ARIA-E, we would expect that as it resolves, that the symptoms would reduce, if not go away completely. For ARIA-H, the areas of bleeding that happen in the brain are always going to leave kind of like a scar or a bruise, and that will always be present. So the MRI is not going to get better from that standpoint. It will always be visible. The symptoms may persist if the bleeding is significant enough that it causes something akin to a hemorrhagic stroke. A lot of times for hemorrhagic strokes, there is kind of an initial severity of the symptoms that gets better as time goes by and the blood gets reabsorbed, but there can be lingering symptoms. The goal of our program is to try to limit the ARIA-H so that it is unlikely to progress to the point of causing a hemorrhagic stroke.
Okay. Next question comes from Colleen, and Colleen asks, is the 5,000 Medicare copay, assuming that the deductible is met, per infusion or per overall treatment?
That's for 1 year of treatment. So this treatment is 18 months from the clinical trial, so the whole treatment would be a year and a half.
Okay, great. And circling back to the Vindamax question, there was some clarification given that apparently it's a medication prescribed for amyloid, and they said it would help. So I'm not sure if that gives any clarification, but apologies if we don't have an answer for you tonight. It's just not something
Yeah,
I think we're.
I'm afraid I am not aware of that for the treatment of amyloid.
Has there been any follow-up after 18 months of the study of lecanemab? I'm assuming, and feel free to clarify, any follow-up for the participants in that study?
Yes, so there is what's called an open label extension of lecanemab, where all of the patients who were in the clinical study, including those that were on placebo, were offered the chance to continue with therapy, and those that were on placebo were switched to active lecanemab therapy. And I don't know that these data have been published in the medical journals yet, but I have seen presentations of these at meetings. And the slope of the patients that have lecanemab treatment for the entire study, including the first 18 months and the open label extension subsequently, that slope continues to be similar to what it was in the original trial. For patients who were on placebo and then switched to active lecanemab, the slope of their decline changes and improves once they start therapy. So it does seem that lecanemab is having an effect on those patients treated with placebo for 18 months and then starting the new therapy. But there is a continued separation between those two curves, as we saw on the slides. And that suggests that probably the earlier the treatment for the amyloid with the anti-amyloid therapy, the better off you're going to be in terms of longer term outcomes.
Right. Is there any way, or how would you limit the risk for ARIA-H?
So one of the things that we do is to try to screen patients before giving lecanemab, and that would be to make sure that if patients are prone to have bleeding, that we don't give the therapy. I spoke about the ApoE gene and how one of the versions of that gene can increase the risk for developing ARIA. And for patients who have that particular highest risk genetic profile, we have a more strict cutoff for the MRI findings so that there's less risk for bleeding to happen in those patients. And then we are requiring that patients not be on blood thinners. In the lecanemab trial, blood thinners actually were allowed, but we are not allowing those to try to reduce the risk of bleeding, and especially areas of bleeding that get worse after they start.
All right. I'll still open up to any questions that folks may have. I do have one question for you, John, and I don't know if this has been something on anybody else's mind, but I think the context being that, we've known about Alzheimer's disease for a long time, several decades at this point. One of the things that I hear often in the work that, we do in the clinic is that, we've known about the disease for a long time. Treatments only up until recently have been limited. Um, there have been certainly, some frontline medications that are often prescribed, but, lecanemab seems to be the first medication that's been introduced that is actually changing the way the disease operates, right? Can you speak at all in terms of why this disease in particular has been so tough to sort of crack and, in terms of this sort of, really important progress in research? Like, why has it taken this long for us to just reach this particular medication?
Yeah, so there are probably several reasons that it has taken so long for the therapy to become available. And it has to do with um, kind of the challenges of studying brain diseases in particular, and also those that have an age factor to them. Um, so because there's very few chances to examine a person's brain directly, the times are either at biopsy or at autopsy. We really don't have the chance to study the brains of patients who have experienced Alzheimer's disease over a longitudinal course in many cases. And this is different, for example, from cancer, where one of the main therapies of cancer is to cut out the tumor, and then that tumor can be studied. Um, so we're sort of, behind the curve because it's so hard to study directly what's happening in the brain. And it took advances in diagnostic testing to be able to identify patients who had probably amyloid or tau in their brain through spinal tap analysis or PET scans. And it's only been within the past decade or two that those tests first were developed and then became clinically available. And even in the early trials of anti-amyloid therapy, they didn't do amyloid PETs. And they subsequently, did autopsy studies when, the patients ultimately passed away, and they discovered that something like 20% of patients didn't even have Alzheimer's disease in the study. So that really messes up the ability to determine how effective a medication is. So being able to do things like amyloid PET and tau PET scans that were done in the donanemab trial really help us understand the biology of the disease in life in ways that up until recently we could not. Another challenge is that in many cases, animal models are used, or cell culture models are used to understand basics of disease processes. And the brain is very complex. It's hard to model in a cell culture system as opposed to growing tumor cells from lung cancer or something like that in a dish and seeing if you put drugs on it, does it stop growing? There are many things that are involved in brain connection between nerve cells, the other support cells in the brain that contribute to its function, that are hard to model in a dish. And then in animal models, such as mouse models, it's hard to compare a mouse that lives 2 years to a human that lives 80 or 90 years. And even with kind of the best models of Alzheimer's disease in animal models, it doesn't fully reflect what happens in the brain of people. So a good example of that is I mentioned in the amyloid cascade hypothesis that there are some rare conditions where there are early onset Alzheimer's disease in families. It's called familial Alzheimer's disease, that's caused by specific genetic mutations in certain genes. And people have taken those mutations, put them into mouse models, and actually cause the amyloid to develop and deposit in the brain. So it can model that aspect. But in those mouse models, without any other manipulation, there's no accumulation of tau. So there has to be other, very artificial manipulations to try to get the tau to aggregate there as well. It probably is an imperfect representation of Alzheimer's disease. So being able to study these things in the mouse model, that is sometimes helpful for understanding disease processes, and also testing drugs is suboptimal in that setting as well. So these factors really make it a complicated disease to study.
Based on that, would you say that as advancements in imaging and early detection improve, so too will the pace at which we hopefully develop new treatments and further our, progress in that area?
That's my hope. And I think, one of the big things that this type of testing is going to help with is making sure that the right patients are getting enrolled in the clinical trials to reflect the disease process that we're trying to treat. And that's going to make it easier to identify the effect that the treatment is having, because we're having kind of a pure population of what we're trying to study rather than having all of this noise from other conditions that kind of mimic Alzheimer's disease in there.
Gotcha. Thank you. Bit of a long question, but I thought it was an important one to ask. So again, still have some time for some additional questions, if anybody is interested in typing them in the chat. The last couple we have on here, I'm gonna go first to one asked by Bob. So is the MRI is for detecting amyloid plaque, but PET is needed to detect tau? I guess they're looking for clarification in terms of which test does what.
Mm-hmm. So there are two different types of PET scans: an amyloid PET that looks specifically at the amyloid proteins, and a tau PET that looks specifically at the tau proteins. And they would have to be done separately. It couldn't be done, together at the same time. It would have to be done on two separate occasions. The MRI itself does not let us see the proteins that accumulate. We can see kind of the effects that they have, such as causing shrinkage of different regions of the brain. The MRI also lets us see if there's areas of bleeding or swelling, but the MRI does not let us know about the tau or amyloid itself.
Okay. Ash has a question here, and I'm gonna do my best to read it here in a way that I think I can get through. So in regards to the 12 and 17% ARIA E and H statistics, I'm assuming for the lecanemab study, please feel free to clarify. Does that study include people who had the APOE gene, or did it exclude them? She's wondering if we have statistics on the percentage of side effects for folks not on blood thinners and folks who do not carry that particular APOE gene.
Yeah, so the figures that I quoted were kind of the aggregate, but both the lecanemab and the donanemab trials broke down based off of the different types of ApoE genes and gene variants. And they can be broken down into sort of like low risk, medium risk, and high risk categories based off of the genetics. And it does provide, the clinical trial data does provide information about each of those subcategories. And it is kind of as you would expect that the lowest risk category has the lowest rates of both ARIA-E and ARIA-H. And the highest risk category has the highest numbers for both. In a lot of cases, the low and medium groups are closer to each other, and the high risk is kind of like far away in terms of the risk. So the highest risk category is really at high risk for developing ARIA. In terms of information about patients who were not, or who were on anticoagulation versus those who were not, I don't believe that is broken out in the clinical trials, so I don't have information about that from the studies.
Okay. Bob asks, is the presentation available for download? Yes, so we are recording this presentation, just so you know, and we will be posting it on the Dementia Care Collaborative website, which I believe was posted before, but I'm sure we can post it again just so everybody has it. So the information and the discussion tonight will all be available for viewing however many times you want on our website. So still has a little bit of time for any other questions if anybody has any burning ones. But otherwise, unless somebody has anything last minute, I just wanted to say, Dr. Dickson, thank you so much for joining us tonight. It was a great presentation, a lot of good information I think will be very useful for everybody who's interested and concerned. Ash, yes. So Ash just wants to clarify, is she correct in understanding that if somebody meets your criteria for lecanemab treatment, their risk for ARIA would be less than the 12 and 17%?
Not necessarily, because we are allowing patients with all of the different gene variants to enter the trial or enter the treatment program. The possible exception would be the highest risk group, because we have more strict criteria on the MRI findings for that. So I would imagine that the risk might be somewhat lower in that group potentially. But otherwise, for the low and medium risk, it's probably going to be about the same, because we're following pretty closely a lot of the criteria that was involved in the trial eligibility.
I just saw that Judy just posted the link for the Dementia Care Collaborative. I will also just post it here. And that's the clickable one if anybody is interested. So hopefully that helps. But again, Dr. Dickson, thank you so much for joining us tonight. Information was wonderful and very, I think, important for everybody here who's interested. Just so that everybody is aware, the Dementia Care Collaborative, our next event is going to be on May 7th, our Health and Resiliency Program with Dr. Felipe Jain, the topic being Restore Your Life Course While Caregiving. So if anybody's interested, please feel free to join us on May 7th for that presentation. So with that, again, Dr. Dickson, thank you so much for joining us, and I hope everyone here has a wonderful rest of their evening. Thank you.
Thank you. Bye-bye.